We now know what DNA is. It's a two-stranded polymer of nucleotides, and each strand has a backbone made of identical sugar and phosphate groups, with different nitrogenous bases pointing inwards, pairing in base-specific manner, A with T and C with G. These long molecules are coiled around histones and then supercoiled to give compact chromosomes, each with many millions of base pairs. All of your genetic material, 23 pairs of chromosomes to be exact, is present in every single cell in your body. But your cells are dividing all the time. Old ones die and new ones are generated to take their place. In fact, apart from female egg cells, there's not a single cell in your body that was there when you were born. So how does each new cell get all of the genetic material? As it happens, all of it is copied through a process called DNA replication, so that when a cell divides, each resulting cell keeps a copy of all of your chromosomes. So how does DNA replication work? It's an impressive operation with about a dozen enzymes working in tandem. Let's look at a few of these enzymes and see what they do. Helicase is an enzyme that unwinds the double helix and disrupts the hydrogen bonds between the bases, thus separating DNA into individual strands and creating a replication fork. The unwinding of the helix generates strain further ahead in the chain, so as we go, topoisomerase will break, untwist, and reconnect the DNA, always ahead of the replication fork. With the strands separate, we can begin to copy each one, but the enzyme that copies the strand needs a place to start. So an enzyme called primase will anneal an RNA primer at a specific location to kickstart the replication. This primer is about 5 to 10 nucleotides long. Then another enzyme called DNA polymerase III binds to the primer and begins to generate a whole new complementary strand, adding nucleotides to the new chain that was initiated by the primer. Nucleotides enter the enzyme's active site, and polymerase catalyzes formation of the phosphodiester bond that joins each new nucleotide as it is added to the complementary strand. This process will be different for each strand because polymerase will always add nucleotides to the 3' end of the existing strand, not the 5' end. And the strands are anti-parallel, so the direction of replication must be in opposite directions for the opposing strands. On the leading strand, DNA replication moves along with the replication fork, continuously synthesizing the complementary strand and requiring only the initial primer. But on the lagging strand, polymerase has to go one chunk at a time, as new template is made available. These chunks are called Okazaki fragments, and they are around 100 to 200 nucleotides long. Each one will require its own primer in order for polymerase to bind and copy the new fragment. After each fragment is synthesized, DNA polymerase I will go through and replace the RNA nucleotides from the primer with DNA nucleotides to make sure it's DNA all the way through. Lastly, because polymerase can't join the last nucleotide of one fragment to the first nucleotide of another, a separate enzyme called ligase has to go through and make sure everything is connected. So to summarize, helicase unwinds and separates the DNA into two strands. Primase anneals primers to start things off, and polymerase III copies each strand. On the leading strand, we need just one primer, and everything goes continuously. On the lagging strand, we need a primer for each Okazaki fragment. Then, polymerase I replaces the primers with DNA nucleotides, and ligase seals everything up. Boom, two identical copies of the original DNA molecule. This whole process, which is happening in billions of cells in your body at this very moment, goes very fast, about 50 base pairs per second. Moreover, polymerase is very good at getting the code right. It almost always puts the correct base across from the template strand, and when it makes a mistake, it can usually backtrack and correct it in a process called proofreading. Even with this, around 1 in every 10 billion base pairs, an error ends up in the final sequence. Luckily, there are enzymes that can recognize these errors and perform mismatch repair, swapping out the incorrect base for the correct one, just like other enzymes that repair damage caused by external sources. These enzymes minimize the possibility of mutation, and we will learn about them later. But, as we said, polymerase almost always gets it right. In this way, each strand in the double helix acts as the template for its complement, and we end up with two identical copies of all the genetic material. When a cell divides, each new cell retains one of these copies, and when the cell cycle gets to a certain point, these new cells go about copying everything again to be ready for another division when the time comes.